Aim. To understand the structural components and their functions of a CO₂, carbon dioxide, incubator. Apparatus/instrument. CO₂ incubator, culture vessels, flasks, plates, temperature, and CO₂ sensors, humidifying tray. Theory. A CO₂ incubator is a specialized laboratory device used for the growth and maintenance of mammalian cell cultures under controlled temperature, humidity, and CO₂ levels. It provides a stable and sterile environment that mimics the conditions of the human body, usually 37 degrees Celsius temperature, 5% CO₂, and 95% relative humidity. The CO₂ helps maintain the pH balance of the cell culture medium through a bicarbonate buffering system. Structural components and their functions. Component. Function. Outer body, cabinet. Provides protection and insulation, made of stainless steel to resist corrosion. Inner chamber. Maintains uniform temperature and humidity for cell culture growth. Door, inner and outer. Outer door insulates, inner glass door allows viewing without disturbing internal conditions. Heating system. Maintains constant temperature, usually 37 degrees Celsius. CO₂ sensor and controller. Monitors and regulates CO₂ concentration inside the chamber. Gas inlet and filter. Allows CO₂ gas to enter and purifies it to maintain sterility. Humidity tray. Contains sterile water to maintain humidity and prevent media evaporation. Fan or air circulator. Ensures even distribution of temperature, humidity, and CO₂. Display and control panel. Digital interface to set and monitor temperature, humidity, and CO₂ levels. Alarm system. Alerts for deviations in set parameters, temperature or CO₂ fluctuations. Principle. The CO₂ incubator works on the principle of maintaining controlled atmospheric conditions essential for mammalian cell growth, primarily constant temperature, humidity, and CO₂ levels. The bicarbonate buffer system in culture media interacts with CO₂ to maintain physiological pH, 7.2 to 7.4. Procedure. Switch on the CO₂ incubator and allow it to reach the set parameters, 37 degrees Celsius, 5% CO₂, 95% humidity. Observe the temperature, humidity, and CO₂ control system on the display panel. Open the door carefully and note the internal components, shelves, humidity tray, CO₂ inlet, sensors. Study the function of each component as explained by the instructor. Record observations. Observation. The CO₂ incubator maintains optimum conditions, 37 degrees Celsius, 5% CO₂, and high humidity, for the growth of mammalian cells in vitro. Result/inference. The CO₂ incubator effectively provides a controlled environment suitable for the growth and maintenance of animal cell cultures, ensuring proper pH and sterility. Conclusion. The structure and components of a CO₂ incubator help maintain precise physiological conditions essential for cell culture experiments in biotechnology, microbiology, and biomedical research. Precautions. Do not open the door frequently, it disturbs internal conditions. Use sterile distilled water in the humidity tray. Ensure CO₂ gas cylinders are properly connected and leak-free. Regularly clean and disinfect the inner chamber. Avoid overloading the chamber with culture vessels.